In this video, we will be investigating the question of why TNF-alpha impairs peripheral glucose uptake. We will first define the problem in terms of conservation of mass. We will then form our hypothesis based on an analysis of studies done by Stevens and Pekala and Plumgaard. In terms of conservation of mass, we looked at blood glucose as the mass, with glucose as the inflow and peripheral glucose uptake as the outflow. With infusion of TNF-alpha, the outflow decreases as the inflow stays constant. Therefore, blood glucose, our mass, decreases.
In regards to blood glucose concentration, we hypothesized that in the presence of TNF-alpha, peripheral glucose uptake decreases due to a decrease in GLUT4 translocation to the membrane. Therefore, a decrease in outflow is seen, ultimately decreasing blood glucose concentrations.
In a healthy control subject, a constant infusion of glucose increases insulin production, activating the insulin signaling cascade. This causes the translocation of GLUT4 transporters to the plasma membrane, increasing glucose uptake into skeletal muscle.
An in vitro study in 1991 by Stevens and Pekala investigated transcriptional repression of GLUT4 genes in adipocytes by TNF-alpha. Recombinant TNF-alpha was provided and 3T3-L1 adipose cells were utilized in this experiment. The pre-adipocytes were cultured, maintained, and differentiated. In order to examine the distribution of GLUT4 protein, Western blot analysis was performed on isolated membrane fractions from control and TNF-alpha treated cells using antipeptide glucose transporter antibodies. The treated cells were exposed to 5-nm TNF-alpha. The results of the experiment identified that the exposure of adipocytes to TNF-alpha resulted in the loss of total immunodetectable GLUT4 protein and decreases in plasma and inner membrane GLUT4 fractions. These results indicate that the observed loss of the ability of insulin to activate 2-deoxyglucose transport results primarily from a lack of GLUT4.
The image demonstrates the hybridization of in vitro transcribed RNA to the indicated cDNAs or 3T3-L1 genomic DNA immobilized on nylon membranes. This figure highlights the decrease in appearance of GLUT4 when the adipocyte is exposed to TNF-alpha. Vector DNA was present as a control.
In a study by Plumgaard in 2005, 9 healthy men underwent 2 separate experiments involving a saline or a human recombinant TNF-alpha infusion. Before the TNF-alpha infusion, glucose was administered to allow for the determination of glucose turnover. TNF-alpha was found to inhibit whole-body glucose uptake. Importantly, hepatic glucose production was unaltered by TNF-alpha infusion, demonstrating a direct effect of TNF-alpha on peripheral tissues. Using a polyclonal affinity purified antibody that recognizes AKT phosphorylation motif peptide sequences, we show that TNF-alpha infusion impairs insulin action of AS160. Reduced AS160 phosphorylation may provide a mechanism for TNF-alpha-induced insulin resistance on glucose uptake because GLUT4 exocytosis is dependent on AS160 phosphorylation.
Essentially, these studies indicate that there is a decrease in GLUT4 translocation to the membrane. As suggested by Stevens and Pekala, translocation of GLUT4 is inhibited by TNF-alpha. The study by Plumgaard shows that the decrease in translocation may be linked to reduced AS160 phosphorylation by TNF-alpha, which was indicated by a decrease in AKT phosphorylation in response to TNF-alpha.
The 2 studies that we reviewed support our hypothesis in that peripheral glucose uptake would be decreased in individuals receiving an infusion of TNF-alpha. Stevens and Pekala showed that exposure to TNF-alpha resulted in decreased GLUT4 translocation and GLUT4 at the plasma membrane, causing a decrease in peripheral glucose uptake. Plumgaard demonstrated impaired insulin action of AS160 with exposure to TNF-alpha, also causing a decrease in peripheral glucose uptake.
From lecture, we know that TNF-alpha acts as an inflammatory cytokine within the human body. The 2 studies investigated in this video have shown mechanisms by which TNF-alpha decreases peripheral glucose uptake. This decrease in peripheral glucose uptake correlates to a decrease in outflow according to the conservation of mass model. Eventually, exposure to TNF alpha will cause insulin insensitivity and lead to insulin resistance, contributing to development of type 2 diabetes.